Hi, so my name is Alfredo Chavez-Arroyo, and I'm a postdoc in the Baumler Lab at UC Davis School of Medicine. And my affiliation is in the Microbiome Pillar for pig paradigm. And I'm studying how the fecal microbiota of weanling piglets influences susceptibility to infection by ETEC as piglets are weaned. So what we're doing is we're isolating fecal microbiotas from weanling piglets throughout the weaning process. And we're using these microbiotas to implant them into germ-free mice that we breed in our facilities here. We then give these mice our fecal microbiotas from piglets, and we can study how this microbiota develops inside the piglets. And then we can ask how this particular microbiota makes these piglets either resistant or susceptible to a challenge by ETEC. So then we can take our germ-free mice that have been colonized, we can challenge them with ETEC in our facilities, and assess which microbiotas provide a protective role or a non-protective role within these particular mice. And we hope that these studies can then translate into the biology of piglets on the farm, so we can understand how their microbiota may or may not be susceptible. So right now we've completed our pilot study where we've transferred microbiota from the weanling piglets into germ-free mice. We've challenged them with ETEC, and what we're doing is looking at the microbial composition of the piglets in the mice and assessing which particular type of microbial profile correlates with resistance to ETEC infection. We're hoping that this can translate into pigs because, well, first of all, we want to repeat these experiments because we think we have different microbiotas that might be protective versus other microbiotas that seem non-protective. Once we repeat these experiments and validate that, we also want to do further analysis on these microbiotas and further types of infection models. And what we're hoping is that we can better understand what kind of microbiotas are beneficial for the piglets, so that maybe we can develop certain type of microbiotas, or perhaps some type of interventions that we can modify the microbiotas, or perhaps modify the host so that it recapitulates what we see that is being done with those microbiotas to make the pigs more resistant to the infection. So in that way, we think that if we get a better understanding of the mechanistics of the particular microbiotas that might be protective, we think that that might help us develop something that either provide an intervention or even something preventative. They are totally germ-free, they have no bacteria or fungi, and they breed inside these small isolators. Here you have 3 female mice that were born from germ-free mice, and these are also germ-free.